horizontal gene transfer in which bacterial genes can be transmitted between one another by viruses, or in this particular case, bacteriophages. Bacteriophages can carry out the lytic cycle in which the host cell is destroyed or viral DNA can integrate into the host genome to become a latent prophage, and that's what's illustrated here. So on the left, you see the lytic cycle. On the right, you see the lysogenic cycle. So in this particular case, a phage injects its double-stranded DNA in red here into the recipient. The double-stranded DNA, usually within a minute or two, starts to encode a nuclease that will chop up the host DNA. And then rapidly thereafter, the virus will start encoding pieces of itself that will then assemble into phage particles and package viral DNA. Eventually, towards the end of the lytic cycle, the viruses start to encode massive amounts of lytic enzymes that will destroy the bacterial host from the inside out, releasing phage into the world. And then over here in the lysogenic cycle, the virus injects its genetic information, and the genetic information can actually integrate, and it's usually integrating in a very specific place in between very specific flanking DNA. The cool thing about being a prophage in this particular case, or an integrated piece of DNA, is that the virus doesn't have to do any more work anymore. It just stably replicates along with the host and satisfies its goal of persisting. Generalized transduction is a process whereby viruses just sort of package random pieces of their host, and that's illustrated in this little flow here. In this particular case, you've got a phage that injects its genetic information into a His plus, Lys plus host. And so again, the His plus refers to the bacterium has the genes necessary for histidine biosynthesis, and Lys plus just means that the bacterium has the genes necessary for lysine biosynthesis. So as these phage replicate themselves and cut the host into pieces, for whatever reason, in this hypothetical example, the His plus gene has remained intact. And as the phage package themselves, or package their own DNA, and send those particles out into the world, some of the phages are packaged with the His plus gene. And so when those viruses carrying that His plus gene infect some other susceptible host, in this particular case, the susceptible host is His minus, that His plus phage DNA can integrate into the genome and present itself as a His plus genome from now on. And so this particular this particular bacterium actually benefits from this generalized transduction, where it's now acquired the ability to synthesize histidine. So now if the organism finds itself in an environment lacking histidine, it has the capacity to make histidine as opposed to not being able to grow. So generalized transduction can manifest in many different ways, but in this particular flow here, what you're seeing is that the bacteriophage, in this particular case, imagine this phage is packaged with a very deadly toxin, in this case Shigella toxin, and so this phage infects a commensal E. coli strain and transduces it into a Shiga toxin producing pathogenic E. coli strain, which is what we believe happened in the evolution of EHEC, or enterohemorrhagic E. coli, the O157H7 strain that produces bloody diarrhea and renal failure in some patients that are infected with it. And that was presumed to have arose from a bacteriophage that infected a Shigella cell, for example, picked up the Shiga toxin gene, and then transferred it to a harmless commensal E. coli. In specialized transduction, is a little bit more sophisticated. In this particular case, it's not just randomly picking up genes. The virus is actually accidentally taking specific pieces of the bacterial host genome with it. And this typically occurs in phages that are lysogenic, that insert their DNA in very specific places. And so this slide illustrates that point. So here you see I can make it a little bigger. This particular phage has inserted its genetic information, and now it's a prophage. And the insertion is, as I said, usually between very specific flanking DNA. And so something has induced this phage to deintegrate. Usually the induction is some kind of a stress. Like, for example, in the case of when we're infected with a herpes virus, a stress could be getting sick with something else, or being stressed about exams, and suddenly you have a cold sore, and that results in a deintegration and replication, or a lytic cycle. So in bacteria, typically induction means exposure to UV, for example, and so this prophage DNA is silenced because some repressor proteins will sit on this DNA and keep it as a prophage. Excess UV could make the protein fall off and then instigate this deintegration process. And so as the phage is deintegrating, it takes with it a bit of flanking DNA. And if that flanking DNA has some useful information on it, the virus will go out into the world with that flanking DNA, infect something else, and inject that into a new recipient, which can then integrate that into its genome. And now the new host will have that bit of flanking DNA that's indicated here. So bacteria are not without any defenses in these particular cases. They have restriction modification systems. And so in this particular, in cartoon A here, the bacterium is in the process of receiving a piece of DNA. And so restriction enzymes or restriction endonucleases will cut that at very specific sites. And depending on whether or not the integrating DNA has these specific chi sequences that sort of mark it as quote-unquote friendly, Rec B, C, and D will either behave as a nuclease or as a helicase that facilitates recombination. So in this particular case, the incoming DNA has chi sequences on it, which marks it as sort of friendly. In that particular case, the Rec BCD complex will facilitate integration using those chi sequences. And these chi sequences, for example, E. coli has thousands of these things in its genome. So if another E. coli picks up one of those, there's a good chance it'll have a chi sequence on it, and there's a good chance that the DNA will be homologously recombined. These are methyltransferases that methylate DNA at very specific places to prevent restriction endonucleases from cutting the DNA. Over here, though, this is the bad situation in which a phage is injecting its genetic information into the recipient. But the phage, in this particular case, does not have a chi sequence. And as a result, the restriction endonucleases, they'll find their restriction sites, and Rec B, C, and D won't find chi sequences. And so this will immediately go into endonuclease activity and chop that incoming DNA up so that there's no integration and no formation of prophage. CRISPR is another example of a bacterial immune response, if you will. And in this particular case, you're looking at a DNA virus here injecting its genetic information into a prokaryotic cell. And so that's stage 1, foreign DNA acquisition. So here's what we call a protospacer, a hunk of DNA that's been cut by Cas proteins. This protospacer would then be integrated into the CRISPR locus. Here, this thing is integrated, and then they represent it here as this red box in front of a repeat that's in turn in front of the most terminal leader sequence. So you have a leader sequence, a repeat, and a spacer. And then the Cas system will then synthesize a new repeat to the right of that spacer, and then you'll end up restoring the repeat spacer pattern. If new phages infect and inject their genetic information, you'll get CRISPR locus transcription, and so it'll look something like this. Here's your pre-CRISPR RNAs. Cas proteins will cut these guys at specific places to produce the CRISPR, the mature CRISPR RNAs. These mature CRISPR RNAs will then associate with RNA guide proteins and nucleases. And so when these things detect incoming DNA, they can form hybrids, and these double-stranded hybrids then mark the incoming DNA for destruction.